Okay, I guess I'll start. So, so first of all, welcome. We have our dean and a department chair and an associate dean, a former associate dean, um, five PhD students who I was either the advisor or the co-advisor, and two of them are now department chairs. So seems that they've done pretty well. And all of them were co-advised either by by Dr. Sun. So let's after maybe you saw my LinkedIn post about all this, and after I posted it, several people sent me a message saying, "So sorry that you lost your research partner." So let's just start with saying, well, she was my wife and my research partner, and she was an amazing woman, extremely successful, by many measures more successful than me. She was the president of the Digital Government Society. She was the program chair of the Digital Government Conference. She was just in May, she was program chair of the FLAIRS conference in Florida. So she left a big imprint. She also had more citations than me. She studied, she was a professor much shorter than me. So the other thing everybody asked me recently is, how did you meet? Well, I came from Austria to be a foreign student in Buffalo, and she came from Korea, and we met at the foreign student orientation. So things went very, very fast. That was 43 years ago, almost exactly. So even though very different people, but we had a few things in common, and one of those things was a strong belief in education. And she actually got two master's degrees in Buffalo, one in linguistics and one in computer science. She was intensively competitive. She started in linguistics, and when she saw me doing computer science, she said, I can do that too. So she got it, and then we moved to New Jersey, and I interrupted her PhD studies because she followed me as and then we had two beautiful children, and that interrupted the studies even more, if you can imagine that. But then she picked up again and went to get an MBA at Rutgers and a PhD in information management, information systems, really. So plus her bachelor's degree in English education. So she had five degrees, and I think that shows my belief in the importance of studying. I think if you need some kind of a role model, there she is. Now, one of the well, there were more things. Fulbright scholar, and she was also very courageous. She traveled to places that I refused to travel to, like Uzbekistan. We went for the Digital Government Conference in Taiwan last summer, where I reluctantly came with her because this was after a magnitude 8 earthquake with Chinese fighter jets flying around the island. So I was a little nervous, but I actually had a very good experience. I quite liked the place. So, yeah. Unfortunately, the last couple of months were very, very difficult. She was in, and yeah, okay, let's not go too much into this. She was also an excellent cook in 5 different cuisines, more or less, starting from her country, my country, American. She was also a very beautiful woman, if you haven't noticed that. And yeah, I'm recovered, but it was extremely difficult, and I know from experience it will be difficult for months to come. If you're 44 years with somebody and then that person suddenly not there, then you're very, the whole life routine has to change. Now, going back to the idea that she was so much into education, when my son had a little bit of a crisis, didn't want to go, she became the tiger mom and kept guiding him. And she had the to see our son at his commencement online, of course. She wasn't able to travel anymore. And that was very recently, but she lived to see him walking at the commencement. Now let me tell you a story about him. When he was, 15, he swore he was not going to do what mom and dad do, this computer science stuff. So I told him, if I were young, I would do microbiology. And he actually listened to me and got degrees at McGill University, tuition half of what it is in the USA, and Princeton. And then for a while he didn't want to study, but eventually he did, and he got his PhD. And guess what he's doing in his PhD work? Computational biology. So he's exactly at the computer science that he didn't want, and he's now much better in Linux than I am. So if I need advice on that, when I go to him. So I also tried to convince him to get a professor job. He refused that. So he's working at the startup, and now he's going to tell you about his work in the startup. Please welcome Dr. Alexander Geller.
Thank you. Give me a second. There's a cup of coffee here. I don't know if that's beautiful.
You have to answer to me.
Okay.
There's also my phone there. Okay, where's that one?
Okay. Great. Okay, thanks for introducing and embarrassing me. That's usually doesn't happen in a seminar, but I assume maybe it does. So I'm going to start with some numbers. So today I'm going to talk about a 50-year-old grand challenge that was solved, which is that of protein folding. Excuse me. So let's look at some numbers just to get an impression of what we're talking about, okay? The paper, which caused the breakthrough in the protein folding problem, I'll describe a little bit more of the details, don't worry, but just so you know, some impression, over 40,000 citations only within a few years. This is astronomical numbers. 2024 Nobel Prize in Chemistry, can't really go any higher than that, handed out for this breakthrough. And for the industry people, the spin out companies from these technologies, they're worth billions and they haven't done much yet. So they're worth billions of dollars. Pharma companies are throwing hundreds of millions of dollars at them. And they're even saying in their press statements, we don't need this money, but I guess we'll take it. That's how much money they have. So it's a big deal. Just want to start with that. And I think that in academia, you probably see a lot of times people claim revolution happens a lot, like, oh, this paper is revolutionary, this technology is revolutionary. This one's for real. This is a real one. It's a true revolution with effects both on science and engineering. So that's what I want to talk about today in two parts. How protein folding being solved computationally has affected both science and engineering. But just because of the crowd, we're sort of in a data science is sort of broad. I'm going to give a little bit of background, and I would be remiss to not start with indexing 0 in the computing college. So we will start with a bit of history and context for the non-biologists, which is, I think, everyone except maybe me and one other person. I don't know. So biologists weren't treated very well, and other scientists in general, but you can see it sort of summed up by this statement by Ernest Rutherford, very famous, also Nobel Prize-winning physicist. All science is either physics or stamp collecting. That was his contention, right? So, to be fair, for a while he was sort of right. This was what biology looked like about 100 years ago, not even maybe 120 years ago, not even that long ago. Biologists literally going around and looking for new species with a net. And they made beautiful pictures, but it was very qualitative. So I understand why maybe it's considered stamp collecting. Everything changed with the discovery of DNA being the genetic material and DNA sequencing. So Fred Sanger won a Nobel Prize, his second one. It's a bit of a show off. To take DNA from just extracting it from an organism, in this case, it's sort of a virus, and getting the letters of the DNA out onto paper, A's, T's, G's, and C's. Those are the letters of the DNA, A, T, G, and C, and was able to sequence them. But it was very painstaking and expensive. Luckily, thanks to increases in productivity, economic growth, you can see on the x-axis here time, and then on the y-axis cost. So the cost of sequencing DNA has sort of plummeted. So now we have lots and lots and lots of DNA in the NCBI database, 450,000 species, 1.6 billion nucleotides, which are the A's, T's, G's, and C's. So databases are full of just raw, information of the DNA. So we sort of moved from this sort of funny picture of a biologist in an island with a net, sort of kind of guessing what the tree of life looks like. That's there on the left. And now we have sort of a molecular level detail on how all organisms are related, how they're all related, to other animals and plankton and all that stuff. So we have kind of like this really, a move from this stamp collection to a real quantitative and computational biology. So I see it as like this from the Matrix. First Matrix, you haven't seen it, it's a must see for just life. You must see it. But basically, you can see here that he sees the code behind everything that we're living, and we have the same thing now. We can actually just look at the A's, T's, G's, and C's, the DNA of any living organism. So we have it. We have sort of this sort of quantitative computational method now. Now, I would say that was last generation's revolution in a sense, because what we're left with is, like I said, 1.6 billion letters in a database that looks something like this. It's not even readable. You don't understand what you're looking at. This is sort of what the raw data actually looks like. So we have to mine these raw data for insights. And that's really what computational biology and bioinformatics and that field is sort of working on, is trying to mine this information for what are the genes in here and what do they do and how do they relate to each other and what can we figure out just from these pages and pages of essentially plain text files. Okay, that's kind of what we're trying to find out. And that's where the sort of data science aspect of this talk comes in a sense, where we have the genomic revolution and all the things that come downstream of it. So we have this large data set. You could do, quantitative and statistical analysis. You develop tools to kind of analyze it, visualization. I'm going to focus a little bit on how AI and specifically deep learning has kind of really pushed us even more forward. Okay. So if the move from qualitative to quantitative was last generation's revolution, this generation's revolution, which I just mentioned in the beginning with all the money and the accolades and the prizes, is the solving of this 50-year-old grand challenge problem of protein folding. And here are the folks who won the Nobel Prize. I'm assuming most people recognize the guy in the middle as the Google DeepMind guy from AlphaGo, etc. So the big name here that you have to know is AlphaFold. AlphaFold was the reason that all these guys are standing up here and why we're talking and why they had a paper with 30,000 citations is because of AlphaFold. That was really the big breakthrough moment. So I'll talk about in a second what AlphaFold does. That essentially, in one word, it is able to predict how proteins fold. So what am I talking about? What does that mean? So I just told you that we have a database full of A's, C's, G's, and C's, right? And that's DNA. So you can kind of see here a very simplified kind of diagram or cartoon where we have a DNA sequence. And that DNA sequence, again, made of a code of A's, T's, G's, and C's, actually gets transformed. That information is made into a protein, which is a tiny molecular machine, okay, that really does all the work in the cell. So, for example, we have information in the DNA that tells us that this flower is going to be purple. So I can kind of understand that this flower is going to be purple from the information. But the actual mechanism, the thing that actually does the work here, proteins, tiny molecular machines that are encoded in as information in the DNA that fold and become this sort of three-dimensional machine that actually, for example, in this case, make pigments. Okay. Another important thing I should mention, just because it'll come up a lot, are this is a single protein, let's say, that makes these purple pigments, and that's what makes the flower purple. There's also a lot of weight and importance in biology about protein-protein interaction. So you have one protein here and one protein there, and they touch each other. So, for example, we have a very dire example with an immune system cell on the left and a tumor cell on the right, and the interaction between them, between two proteins, one called PD1, PD-L1, names are not important, but they are touching each other, and you can look and understand the molecular details of these folded proteins, how they touch each other exactly mechanistically, like a lock and a key, determines whether this immune system cell is going to kill the tumor cell or not. So interactions and understanding the 3D shapes of how they touch each other is huge also. This is like a life and death scenario. So I said DNA is information that gets transformed into proteins. I sort of grayed out some of the details, but basically, again, what we need to know is that DNA is a code with 4 letters that eventually is sort of translated into this sort of beads on a string. These chemicals, of which there are 20, there's 20 different chemicals. You connect each one based on the information in the DNA as beads in a string. So those first 3 pieces of DNA, so you do it in triplets. Those first 3 letters in the DNA are becoming this amino acid that's called MET. And the second triplet is becoming another amino acid. Okay, so they're encoding information to these tiny chemicals that get connected one after another into a string. Okay, and then these amino acids, we call them, each one of those balls there, beads on a string, is an amino acid. Eventually it folds. So I'll show you again. Here we have the protein, and here you can see on the bottom left that the protein kind of eventually folds into this shape that many times we represent looking like this sort of helices and sheets over here. Okay, so those are the sort of many proteins that have this type of shape. So you can see how this string of beads that was encoded by the information in the DNA becomes sort of this folded shape. And how does it fold? In theory, we kind of understand what's going on, right? So here on the right, we have a string, again, of these amino acids, the building blocks of the proteins. Let's say the black ones here are hydrophobic, fearing water. That means they're fearing water. So chemically, those ones are sort of have this repulsion to water. And the white ones are loving, so they don't mind being next to water. So there must be some configuration in which it collapses, right? So that the black amino acids, which are fearing water, are hiding inside. And the white ones that are water loving are on the outside and can touch water. So that's kind of like what you can imagine how it sort of decides, quote unquote, energetically to fold. Is that you want to get into an energetic state such that the chemical beads, those amino acids that don't like water, let's say, are hiding, and those that like water are showing. So you kind of collapse from string into like a globular shape. Now we get back to the computers. So not a big deal. We have the strings, we understand the code. Why not just do it in the computer, try to understand what the shape of these proteins are? The difficulty here is that every single one of these beads is actually kind of a little chemical with a bunch of different connections to them. And there's angles that connect them. And each one of those has an angle kind of telling you like how they are in relation to one another. And with a very modest protein, you have an astronomical number of combinations that would not be possible to kind of figure out just by sort of randomly trying to fold the protein. Okay, so there was like a name for this, Leventhal's paradox, where it's like it should be sort of computationally, even physically, impossible to find the right shape, but somehow, lucky for us, the proteins fold up okay. Otherwise, we'd be in trouble. So it's very, very difficult computationally to figure out how this strings on a bead amino acid chain becomes a protein. So you can't simply model it just with the physics of it. So that's why, with a little bit of background, you can maybe understand why, again, so much accolades and money were thrown at this, because AlphaFold is actually able to take in that linear amino acid sequence, just the letters themselves, and then output exactly what the structure is meant to be. So here they're, showing a really powerful result where in green, it was like a real protein that was imaged, using the crystal structure technology and the computational prediction in blue. So you can see that AlphaFold really nails exactly what the shape is supposed to be, right? So this is really amazing. And the way that it works, I won't get into all the details, but just I'll highlight some things. Basically, you can see the input is a protein sequence, as I said, the linear sequence, and out comes the 3D structure. That's kind of what we've been talking about. That's the main takeaway if you don't follow everything else. We also get a confidence score. So it tells you not only the shape, but you can see here a little bit the colors. It tells you, okay, this area over here is we're not so sure. This area over here, we're pretty sure. So there's confidence scores. And the way that it essentially works, it sort of came in parallel with the LLM sort of ChatGPT moment, that revolution, because the core of the algorithm actually uses self-attention. But instead of using natural language, it uses the protein language. Like I said, it's a language simply made of 20 letters of one after the other. And similar to natural language, where you kind of have an LLM, which after the model is trained, you have kind of this self-attention mechanism that tells you which words are sort of semantically connected to each other. Here we have the same thing, except we find sort of long-range dependencies. So remember, this is a strings on a bead. Eventually, if it folds, I know that number 1 and number 17 are going to eventually fold up next to each other. So there's sort of a long-range dependency, sort of like the grammar. Like there's a word "it," and then there's a ball, and then it kind of is referring to each other. We have these two, amino acids that are far away from each other in 1D sequence space, but actually close in 3D sequence space. So you sort of build, using self-attention, this kind of long-range dependency map, which can then be made into a structure. So really, at the core of it is the self-attention mechanism. Again, like an analogy to natural language, how you find connections between words. And here we find connections between the amino acids and their positioning in space. Okay. Okay. So that was a whirlwind tour of biology from a net, catching a jellyfish with a net, all the way to AlphaFold. Okay. So that was a quick, hopefully something that got through. So now I'll confuse you even more with some science and engineering. So two main parts of the talk, now that we know a little bit of biology. Number one is science. So what effect did this AlphaFold revolution have on science? I'll talk about a project I did in my PhD. It's published, the reference will be shown later. But basically, I'm starting to work on this really amazing system. So let's just take a moment to marvel at what we're looking at here. There's a bacteria, this tiny microscopic organism that exists. And just like every other organism, you see like in a jungle in a nature documentary, they're fighting each other for survival. Bacteria are no different. Just because they're tiny, micro scale, doesn't mean that they're also not fighting each other, right? Where did we get antibiotics from? A fungus trying to kill all the bacteria, right? So here we have an amazing system called the type 6 secretion system. Okay, I'll be talking about that a lot. It's abbreviated as T6SS. And you can see that it's sort of a molecular machine. I showed you a dinky protein before that makes purple pigment. Look at this machinery. This is a virus that injects DNA into its cells. So the virus has to come in and injects its DNA and replicates itself, just like a sort of a virus on a USB or something like that. It's put in the computer and then it's injected into the computer. We have this physical injection. Somewhere along evolutionary history, bacteria themselves stole this
injection mechanism made of this crazy, each one of those things there is a single protein that kind of comes together and to make this amazing machine that actually contracts, as you can see here, and injects sort of a spiked tip, like almost like a spear, like some sort of spear shooting outwards and actually can hit and stab nearby a bacteria. So you have one bacteria, and here embedded in its membrane, sort of on its outside, is this machinery that can actually literally shoot and stab competitors. So, we're not the only ones, humans are not the only ones fighting each other, and animals are not the only ones fighting each other. We literally have bacteria stabbing each other with these crazy molecular machines. Okay. And it's like I said, a bacterial nano weapon. It punctures target cells. And if it wasn't enough, the tip of the spear is actually has poison on top of it. Okay, like we're very sophisticated engineering here. We have proteins, other proteins. So we have the proteins that are the machine, and we have other proteins which are called effectors. They're usually toxic proteins. Okay, so the tube is filled with effectors. The tip has effectors on them, even sometimes attached, which I'll talk about. And these effectors, you can imagine, maybe they're little. I have them as little Pac-Man. They come in and maybe chew up the DNA of their competitor. So they not only stab and puncture them, but it's almost like a syringe. They deliver these proteins which are so toxic and kill the neighboring cell they're competing with for resources, right? The one thing is just to make sure that you yourself are safe. Let's say the attacking bacteria is safe. There's also immunity proteins. So if I have some Pac-Man that chews up DNA and it's going to destroy all my information, like we don't want to have any corruption of my information inside the cell, we have some other protein that almost covers this mouth of the Pac-Man that prevents it from being toxic, and that's called the immunity protein. Okay, so we have the effectors, which are these toxic proteins that are injected, and we have the immunity proteins, which are binding to and inhibiting these effectors. Okay, so we have that's what we have in the attacking cell. Hopefully in the target cell there's no immunity protein so that the attack will work. Okay. So one of the easiest ways to load these toxic effector proteins onto the system itself to launch is simply by tethering the machinery. So you have the tip and the spike and the tube, and if you want to just load them engineering-wise, it might be the easiest way to load them is to simply attach them genetically. So we add to the end of the gene of the tip, we just add some information about some toxic gene, and that effector code is sort of downstream of the machinery of this weapon, the type 6 secretion system. And so my goal was to sort of look in this vast database that we have of all the DNA ever and look for something that looks like this, something that looks like a tip with a question mark downstream, something that looks like the tube with a question mark downstream. And we can see maybe those question marks, something interesting. We know that many times downstream there's some toxin. We need to get those toxins for developing medicines. Nature's way of finding and killing bacteria is much smarter than us. So if we can just find some of these question marks and determine what they are, then we're going to have new proteins that maybe kill bacteria, and we can use them eventually as medicine. So when you actually look at the tube and the tip and the spike, there are tons, especially within the tip and the spike, which are unknown. We have this sort of signature in the genetics where we have something that looks like that tip of the weapon and something downstream, which we don't know what it is, which is, probably some sort of toxin. So what we're trying to do here is, again, I'm looking for more of these toxic effectors. Like I said, they might have some downstream effect in medicine. In these elongated type 6 secretion cores. So in this elongated cores, I think that there's going to be new toxins. And the methodology here is where we bring in what we talked about before, a bit of AlphaFold, a bit of another program called FoldSeq. I don't have too much time to talk about it. But basically what we're bringing in are these structural bioinformatic tools to help kind of enrich our understanding of what's going on. So what did I do? Basically, I went to the database that has every sequence ever, at least of bacteria and other microbes. And I look again for this signature where I have like the piece of the machinery and downstream sort of question mark. I don't know what it is, but hopefully something interesting.
Then genetically, many times bacteria are very organized, more organized than humans, by the way, where they have the immunity protein, which I said is critical, right? You don't want to be killing yourself by accident. You have the immunity protein exactly encoded right downstream. That's what that green represents. It's like the gene next to you have kind of just to make sure that you don't lose it and you always have it next to you. You have the immunity protein next to it. And the next thing we do basically is use AlphaFold. And this is where we understand whether that gene next door actually binds, right? So effectors and immunity proteins need to bind each other, otherwise they don't work very well, right? Like I showed before, the Pac-Man being covered by this sort of crescent, it blocks its mouth, it prevents it from chewing the DNA. That's the effector, that's the immunity protein. We should be able to use AlphaFold to understand whether a protein binds one another, because AlphaFold understands if you put in a sequence or even multiple sequences, how they are in relation to each other in three-dimensional space. So I mentioned that AlphaFold outputs three-dimensional structure, but it also outputs a confidence metric. Okay. One of those confidence metric is called PAE, the predicted aligned error, that describes confidence in the folded model. So it gives you a 3D model, but it also tells you how confident it is. And basically what you're getting is a matrix. The amino acid coordinates here are on the X and Y axis. So it's like a sort of from amino acid 1 to 100 on the X axis, and on the Y axis from 100 to 1 to 100 from top to bottom. It's a little bit strange. It's not your regular XY coordinate, it's the other way, but it's a matrix of self versus self. And in protein biology, they call N the beginning part of the protein and C the end part of the protein. And so I sort of, you know, these are not the real meanings, but just as a heuristic, N is like sort of the beginning, and then C is like protein completed. That's the end. So N and C, like N is the beginning and C is the end. So what are we looking at? Basically, what we want out of our model, like if the AlphaFold is working, we have a low error, right? Low predicted on error, which means high confidence in blue. If there's a high error, then we're not so sure what's going on here. So we'll go through it a bit slowly, just because I know it's a lot of stuff I just said. But basically, what we have here is that we have the protein, let's say, that was folded. This pink protein was something that was folded. On the X and Y axis is the linear version, 1 to 100, let's say. And here the folded version is like a sort of Pac-Man. So the full protein here, in this case, has a low PAE score. High confidence. So it's really, this is perfect. This is the exact thing you want to see. So if you look at this section of the matrix, we have at residue 1, or like let's say residue 10 and residue 12, we have very high confidence where they are in relation to each other in space. Very high confidence because the spot is blue. So at that spot at residue 1 and residue 12, if you could see also in 3D space, we have a very high confidence of where those are supposed to be distributed next to each other. The same thing here, residue, I don't know, 90 and residue 80, we kind of have like a very high confidence that those residues are aligned in 3D space exactly where they're supposed to be, okay? And same here between residue 10 and residue 80, whatever it is. Now, sometimes we get something funny that looks like this, where we have within this section of the protein, so within residue 10 and 12, we're quite confident that those two bits of the protein are aligned exactly in space as they're supposed to. And sometimes in the residue 80 and 90 are aligned exactly in 3D space as they're supposed to be. But between residue 10 and residue 80, we're not so sure where they're supposed to be. That's why it's red. We're not so sure. So you kind of get this protein, which there's one glob that kind of we're pretty confident, and another one, and then in between them, maybe you can imagine it kind of floating around each other. We don't know where they're supposed to be standing, one next to the other. What's nice about this metric is that it actually expands to protein-protein interactions. So remember, our whole goal here is to kind of predict whether the effector toxin and the immunity protein are binding to each other. So by analogy, we're doing the same thing that we did before, except now I add another protein. So I have on the x-axis my, let's say, effector protein and immunity protein in red and green respectively. So we have the effector in red and we have the immunity protein in green. And the sections of the matrix are basically operate in the same way, where if I'm confident about the binding in whether I'm within or between the proteins. I'll show you in a second. So basically here, this is the beautiful, amazing case where we're super high confidence, but really means very high confidence, right? That the protein is itself folded well, and we know that it's organized relative to each other in 3D space. So here we can see in this section of the matrix that the pink protein itself folded great because everything is blue. In this section of the matrix, we can see that the green parts of the protein relative to each other, we're very confident. And in the plus plus and minus minus quadrants of the graph, we also see that, the pink residue, like pink residue number 7 and blue residue, whatever, number 15, they're perfectly aligned in space exactly as you expect them to be. So they're really in the place where you're high confidence that these two proteins are exactly aligned the way they're meant to be. Now, contrast that to this case, where we have this sort of stripy pink protein, where although the stripy pink protein itself seems like it folds okay, and actually the green triangle protein seems like it actually folds okay, relative to one another, we're not confident that they're supposed to be aligned in space. So you get a scenario where the confidence, although the proteins themselves alone fold okay, relative to one another, we can't understand where they are. Okay, so if you didn't follow the thing I just said, which is totally okay, basically here's the take-home message. We're looking for interaction between our effectors and immunity proteins. So what we're looking for here is a signal in these sort of diagonal quadrants, the plus plus and minus minus. We want to have blue over, and if there's no interaction, it's red. That's the takeaway. Okay, so this is one of the metrics AlphaFold outputs. It's so useful. It's amazing. So I'll focus on 4 effector immunity pairs, just to kind of pare it down, and I'll really only talk about one in detail. And we named them type 6 effector candidate and type 6 immunity candidate. So because we're doing bioinformatics and not necessarily experimentation until the end, we basically call it candidate, but it's like the effector of the immunity in short, E for effector and I for immunity. So let's take a look and let's see if we learned something. Basically, what we're looking at here is the connection between the effector candidate 1 and immunity candidate 1. And what do we see? Well, the green shows you the grid, like where the breakdown of the quadrants are. And so what do we see? We see blue in the plus, plus, minus, minus. So what does it mean? In short, it means the effector and the immunity are predicted according to AlphaFold to bind to one another because we're confident in their positioning in space. Okay? And to contrast that as a control, I sort of looked at, let's start just by looking at this. I shuffled them. I took the first effector, EC1, and T6 IC2 from a completely different organism. Remember, we're taking from a database. One was collected, in a forest in Shanghai, and the other one was collected at the bottom of the ocean. There's no connection to them whatsoever. And so I put them right next to each other, and AlphaFold says they don't bind. We don't see that blue signal in those quadrants. And we see the same thing here, although there's a little noise always. We sort of see that there's no signal for binding. And when I add, and you can see on the diagonal, you really do see that those that are meant to be binding each other, those that are next to each other, have a signal for binding. And as a sort of computational control, you can see that those that are shuffled, which again are completely different bacteria from completely different places in the database, don't have like a very strong signal for binding. Okay, so we're able to use AlphaFold to really confirm and give us confidence that these proteins bind one another, and that can save a lot of money and time in the lab. And this is what they look like. We talked about folded proteins, so you can see kind of the actual shape of them. Like I said, there's going to be a lot of helices and yeah. Sorry, a lot of questions to us. Of course. So basically, you would a different way to connect this, like the two working together, right? Mm-hmm. Find out whether there's any way that they can bind or not binding. Right. Yes. One question is the number of trials that we use for this tool. How you ensure that you have used 4? Yes. Yeah. What was for this? So we did more than 4. So we did a lot. We did like more statistics and kind of understand looking at. We also use kind of control like known binders, known and shuffled. So we have like a model that actually there's the score that we calculate the signal here with a logistic regression with a known data set of binders and then the shuffled data set. So we have two positives and sort of shuffled version of those, which should be negative. And we have a clear signal, like statistically and strongly. I only focus on these four particularly because number one, they were novel, so they were not known before. And number two, we just did more experiments on these. So it was from a practical measure. We didn't by any means just do four. We did like a more comprehensive model to, yeah.
Since you found the experimental results,
Yeah. So some of these have nice experimental results, so we highlight them in talks and papers. That's the yeah.
Thank you.
Sure, of course. So basically, here the structures of those four that I talked about, the effectors and the immunities. The effectors are in red. That's what they actually look like, and the immunities in green. And another little tidbit that just shows that I think that not only is the score, I mean, I trust AlphaFold, that I already do, but you can see that some important amino acids in yellow, I won't get into it below, but you can see that really these green proteins are seriously blocking exactly where those important amino acids are. So there's this red protein which does some, like I said, these are toxins, they do some sort of crazy, they're enzymes that cut DNA and all this stuff, and these immunity proteins really block those important amino acids that do that function. So another piece of evidence that just shows that this is the right setup. And when I won't do all the details of this graph, but basically in one word, what we see here is that when I put this effector in a cell, it kills the cells as you expect. So again, remember, this is just some question mark. This is something I found in a database sitting on my computer. I didn't see anything. We pulled it out of the computer, make it into a real protein, put it in the bacteria, and the bacteria actually dies. That's what we see here in this sort of lavender purple sort of color. When you just put the effector in the bacteria, it dies. So it kills the bacteria. And then when you actually add the effector and immunity protein, which we predicted with AlphaFold, we know it should bind, it actually saves it. So the higher it is on the graph means the more it lives, which you can see in the sort of green color. I won't do all the details, but in short, basically what we saw in the experiment is that those effectors that was just some question mark in some database that we predicted are toxic, and that the gene next to it, which we predicted in AlphaFold structure, actually does save it from dying. So it's really the experimental proof that the effectors are toxic, and the immunity protein actually presumably binds and inhibits that toxicity. I'll talk briefly about this experiment and then move on to something else. But I'm going to focus in on this specific effector immunity pair. So now that we know that it's toxic, right? So I took this protein out of the database, I predicted with AlphaFold, I know it's toxic, I know the immunity is in immunity. I want to know what it is. It's totally new. Again, it's question mark. So what do? Usually, back in the day, 3 years ago, we take the sequence and we search for similar looking sequences in the giant database, and maybe some of those have some hint as to what it's doing. When I search for this one, there's nothing, zero in the sequence. Well, it's nice, and this is another tool. I don't have much time to talk about it, but please look it up. It's amazing. FoldSeq, which does a very beautiful heuristic search for the structure. So not based on the sequence, but again, using this sort of structural bioinformatics type of revolution, we're able to search the structure and find something where the sequence showed nothing. So the sequence shows nothing. The structure actually finds that our protein, T6EC3, whatever we're calling it, shows a structural similarity to this protein SLEB, a protein that cleaves the bacterial cell wall components. So you can see here in red, here's the protein that we predicted. So PAAR, that's the spike of our machinery. Remember, this is just a giant stabbing machine. The sharp tip of that spike, and connected to it, we have this unknown protein, but we know that it's toxic, and we have the immunity protein sticking directly into it to prevent it from killing itself. And you can see in blue, this thing that we found with the structural search, SLEB, where the sequence search failed, we were able to find this. And SLEB, we know, is filling up the cell wall of bacteria. So we had an idea, if this protein, T6EC3, is similar to this cell wall cleaver, maybe it also cleaves the cell wall. So if we turn that on, it should maybe change the shape of the bacteria. And the reason is because bacteria, like they have this sort of classic rod shape, at least E. coli does, some other shapes exist, but it's almost like a balloon because there's so much pressure inside it could explode, but there's a cell wall that keeps it in place. So you can think of it almost like a balloon with a wire mesh inside of it, outside of it. The wire mesh is holding it in place, and there's so much pressure inside, but it's preventing it from really expanding. But if you remove this wire mesh, then suddenly the balloon can blow up because there's so much pressure. So the bacteria has so much pressure inside, but the cell wall is keeping it into this rod shape. So the idea is we have this protein that looks like something that maybe chews up that cell wall. And so what do? We took regular bacterial cells, and first of all, here we have, I believe this is the just regular bacteria. You can see it has this sort of characteristic sort of rod shape. The colors are from the microscope. Basically, what we have here is the membrane stain, so you can see the edges of the bacteria. In blue, rather, you see the DNA, and then that's just an overlay of the two images. So that's what we're looking at here. We're looking at a microscopic view of these bacteria, and what we see here, amazingly, is when we put this T6EC3 protein inside the bacteria, we get this cell rounding. So we've removed the wire from this balloon, and the balloon is expanding like crazy, and you see that it's losing its shape. So computationally, we predicted using this fold-seq, where the sequence search failed, we found a protein that kind of looks like this one that chews the bacterial cell wall, and we can really see here nice proof that this is in the right direction. We really see that this protein seems to be chewing up the cell wall because we're getting these round cells. And there's a nice video of this happening as well. I know it's a bit chaotic, but you can see cells that are shaped as rods suddenly losing their shape and becoming round. So and eventually exploding, by the way, they eventually die. But you can kind of see these rod-shaped cells, like over here, and then suddenly, boom, you turn on this protein, they become round. So we really see before our eyes in the microscope that the prediction on the computer seems to be really real. So that was a lot, especially for non-biology crowds. So I'll just summarize. We found 4 new effector immunity pairs, amongst others, and we used AlphaFold. And the key where we used AlphaFold was to predict the binding between 2 proteins. So it was AlphaFold with a multimer. It's a specific flavor of AlphaFold. And fold-seq, please look it up. It's a very nice algorithm. Is powerful for annotation as well, because we're able to search for structures and not sequences, which is much more sensitive. Okay, and the paper is at Molecular Systems Biology, if you want to take a look. Okay, so that was part 1. Yes, please.
Will you please elaborate on the potential application of this in real life?
Yes. So of these factors, you mean? Yes. So 2 things. Number 1, we might be able to make sort of antibiotic drugs out of these proteins, since they cure bacteria and they cure them in new ways, maybe that we humans have not been able to engineer. We might be able to develop those into drugs. But if we don't, the second application would be that we could actually use the targets. Sometimes what's interesting more than the actual protein itself is what it's targeting. And we can learn from that target and use that as a target to develop in the pharma industry. So a lot of it has to do with medicine. The second, which I think it really has an effect, is on ecology. So when you're looking at, let's say you take a plant out of the ground and there's roots and there's soil and whatever, in that soil around the plant is a population, like a city, of many different kinds of bacteria and fungi and all kinds of things that are living there, competing there. The plant is releasing a lot of delicious sugar, so they're killing each other to try to be on there. Predicting those dynamics can really have an effect on the health of the plant. Similarly with us in our gut, we have a lot of bacteria. The dynamic between all those bacteria, who is killing who and how, really determines how your gut's going to behave, and that has a lot of effects on your health. So if we can catalog all these weapons and who's using them and who's immune and who's not immune, right? Who has the immunity proteins, who doesn't have the immunity proteins, who's a friend and who's a foe, and put that into some kind of network, we'll have a better understanding of ecology at sort of a molecular level. That would be sort of the grander scientific application. But yeah, those are the 2 main spheres. Yeah.
Yeah. So
Like right now you have to find in the lab a loss in order to see whether the protein is binding with it or not, right?
Yeah, yeah.
I'm wondering there's any tool out there. You are simple, right? And I don't know, build a training model in order to play the game. Like, hey, I gotta put one protein structure here.
Yes.
Go on, like
Yes.
to find to match them,
Yes.
right? So
Oh, yeah.
any tool like that, then it's significantly reduce the time. But
Yes. Agreed.
only you have to in order to combine these things together, right?
100%. So it's a great idea. There's a lot of so we do, although we do, it's okay. So the tools that, for example, use some deep learning type of models to make like a surface fingerprint of interaction faces. So you have two proteins, they interact, and you have sort of this like fingerprint of interaction. So and then you have kind of two data sets, one of one face and one of the other face, and you can kind of connect between them. And that's very much so. Yeah, that would help a lot. You don't have to maybe run a heavy model to do it. Maybe it's easier.
But that I think like we did what you are doing because in order to discover only this type of combinations or categories, categorize them in whatever environments, right? It's got to be tremendous amount of money, time.
Yes.
In.
Yes.
That bottleneck will be, let's say, a tool that can help you to really integrate one with another one. Very effectively, similar to another fold, like AlphaFold, but like it's kind of like I don't know, connect them.
Yeah. 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 That's a good point. I mean, like one of the things we see is that you might work on, you decide, this protein works for me or whatever, and then you take it to the lab, and then it doesn't express, or it's too toxic, or in this bacteria the target doesn't exist, and all these things. So there's a lot of inefficiencies that could be fixed, like with a more combined model or something.
Yeah, it was very simple, right? If you take if you already integrated,
Yeah.
you found that is binding, right?
Yeah.
You take them out and you push them in a way that like you have to ask the model to link them to the bindings.
Yeah.
Like yes, a random state of the two. The probabilistic model replace allows you to really go to the binding state that you need.
Yeah.
Something like that.
Yeah. There's a model that is exactly as you said. There's something that does that exactly as you mentioned. It moves in that case, it's RNA. It doesn't matter. Different molecule moves it, and then if the model can move it back, the confidence in the being is higher. There's a model exactly like that, that you're saying. It's a great idea. So you already nailed it. Yeah. So yeah, sure. Okay. So if there's other questions, I'll move to part 2. So that was science. So in one word, basically, we're saying science essentially can be helped a lot with these structural algorithms, whether it's for predicting binding, for annotation, and for a million other things. This is just one tiny sliver of microbiology that's is affected by this, but medicine is affected by this. So now I'll talk about my industry experience. I just started. It's basically, I work in a startup called Denovai. You can see the name right here. What we do is protein design using both the AI that I'm talking about right now and also physics-based methods. So we kind of have a combination going on. And what's nice about this startup is that it's within what's called a venture studio. So the venture studio is called Ion Labs. That's the big name there. And that's me with the long hair. Basically, the venture studio is like an incubator, but the venture studio decides ahead of time what the startups are going to be. So the venture studio, they talk to their pharma partners. So we work with Teva, Pfizer, AstraZeneca, and Merck. They're sort of these slower bureaucracies. So they say, what type of innovation do you need? And we'll make a startup for them. So the venture studio, Ion Labs, they talk to the pharma partners and they say, okay, you need protein engineering. So they make a challenge to academics, industry people, and whatever, and whoever wins the challenge, they start the company. So it's almost like a startup farm. There's a problem, and then they sort of put water on the startup that's going to solve that problem. Okay. And so one of our companies is within this umbrella here. And again, what we do is protein design using hybrids of AI and physics methods. So one of the technologies that we are licensing is actually directly related to us, like actually is made by our CEO, our consultants at the Ember European Molecular Biology Laboratory. It's called Alpha Design. So it's a framework for designing proteins that's based on AlphaFold. So once AlphaFold came out, basically everyone started going in every direction. Now we're talking about the engineering direction. So the engineering direction is basically how we can make new proteins that didn't exist before in nature ever using AlphaFold. So basically the way that it works, Alpha Design, is protein hallucination, essentially, right? So we put in sequences that are sort of random and never existed in the world. There's a huge sequence base out there that hasn't been explored. And you put this sort of non-existent protein that's never existed before ever into AlphaFold. And out you get these metrics, for example, PAE, which I talked about at length before. So you have some confidence about a protein maybe binding to another protein and how it folds to each other. And then you define some fitness functions. So let's say I want to bind my target. So I have my target floating over here, and I put in a random sequences of A's, C's, G's, and C's, which become the 20-letter alphabet that I wrote into AlphaFold. And then the PAE score is terrible. It's all red. So then what do? Then we iterate with evolutionary algorithm. So there's an algorithm that takes a pool of sequences and does something similar to what evolution does over time, does some mutations, does some recombinations, basically mixing and matching the sequences, changing the sequences, and then tries again to put into AlphaFold. So we have this iterative loop where we're trying to get closer and closer to a PAE matrix, which, like we said, looks blue in the right places, right? So we have a function in which we can kind of optimize for, and we can go with this evolutionary algorithm to get closer and closer, hopefully, to a sequence that actually does what we want. So we can make a protein sequence in 1D that, with letters that never existed before in the whole universe, but actually folds into something that AlphaFold recognizes as, let's say, binding to some target, okay, through this iteration. That's the how we do protein hallucination with Alpha Design, which I think was really something crazy. And it's totally new. This was not this type of thing, but it was a big revolution in the engineering field as well. Actually, part of the Nobel Prize was given for engineering and not just for AlphaFold. A similar method, which I really love as well, was from a tool called Minecraft, which also does protein hallucination. Okay, so what they do, and this is sort of a similar diagram, but I'll highlight the key difference. But what they do is they take that random sequence, they put it into, let's say, AlphaFold, and then they have a prediction. And then they have a target, which then could be, the PAE score, like the score that we're trying to go for. And then they do sort of classic backpropagation where they calculate a loss. So we kind of are way off, let's say, the binder doesn't bind at all and it's not in the right places, but there's a hint in which direction to move. And then they use weight that backpropagates all the way back, not to the weights, because the model is trained that the weights are frozen, the weights and biases are frozen, and it goes all the way back to the input. So the backpropagation all goes all the way to the input, and we get sort of more precise changes in the protein sequence space to get closer and closer to where we need to go with the target. So I think this is a really elegant thing as well. So we use the evolutionary algorithm. This is a really beautiful way of kind of changing the sequence in sequence space just to kind of optimize for, again, let's say, binding. So those are some of the methods for protein hallucination. One of the other bits that is used a lot these days now in the protein world are diffusion models. So again, we talked about AlphaFold sort of being parallel to this LLM revolution. We have also on the image side, right? We have kind of this diffusion models for images. The protein field borrowed that concept also for diffusion of protein. So you see a beautiful image here from like one of the big famous models, RF diffusion. The diffusion model for protein structure. So I'll just briefly mention it. It's very similar to what you see with images, but basically what you start with, and we start here right to left. In this case, we have a protein structure, which I said again looks like these sort of helices and loops and sheets and stuff. And in a stepwise manner, in each step, you add a tiny little bit of noise, and that noise sort of moves the atoms a tiny bit out of the place where they're supposed to be. And then you do another step, it moves a little bit farther away, and another step until you get to sort of total noise, like really like white noise. And what the model learns is essentially to denoise data that's been corrupted with Gaussian noise. So after one step, it's a little bit off, and it learns to kind of put it back to where it's supposed to go. After two steps, it moves back to the previous step. So you learn you train the model by noising it into noise, and then actually, then it's able to kind of hopefully get back to where you want to go. And just like with the image models, the cool thing about it is that if you do random seeding of your Gaussian noise at the end, in a sort of noise state that's never been seen before, what you're going to get when you actually do the inference is you're going to get a natural looking but new protein that you've never seen before. So a diverse output that kind of looks like the training data, right, which are real proteins, but are generated new. So you can actually make new proteins protein-like in every single way. They sort of legally look like proteins, but perhaps have never existed before. And you can do even better if you do conditioning. So in the most simple case of conditioning, you kind of maybe you put a target. So you have this green protein, which is some, let's say, some medical target, something important, and you start with a cloud of kind of pure noise, and then you can actually do the diffusion sort of influence to create a binder conditioned on the existence of this second protein nearby. So you have a diffusion process where the target protein is there, and then the sort of cloud of points coalesces into a protein that sort of looks like a protein and binds this target, right? So we kind of condition it, and there's a million different conditions you could do to sort of guide the diffusion model. So like I said, the application would be, yeah, you can make binders against any target you can imagine, which is for medicine, that's pretty much the whole thing. So already, I'm just sort of doing a broad overview here of some of the things that engineering is using these tools for, like protein hallucination. So like I said, we can use an evolutionary algorithm, we can use backpropagation, we can use diffusion models to kind of like make proteins in every which way, including conditioning it on binding targets. And there's really tremendous potential for medicine, for industry, for nanotechnology, for a million different things. It's really like an exciting field. Things are moving so fast that you can really keep up with open source and industry. Like I said at the beginning, tons of money is being thrown at this. It's just a really cool place to be. So overall take-home message here, AlphaFold is really like huge major breakthrough, a real revolution, not just like what people say. And you can understand now, hopefully, why so much accolades and money are thrown at it. Science-wise, we can kind of really view things from a new angle. In bioinformatics, we see everything from the structural point of view now. It's much richer in information. And for engineering, protein design is so much easier and efficient now. It wasn't really a field as much as it was, but now it's like everyone's doing it, and pharma is all over it. So it's really an amazing place. So that's it for the science. I'll talk briefly also. My dad kind of said most of this stuff, but I did want to just make it clear. I know it's a little bit strange of the chair of the department invites his son for the talk, but I know it raises some eyebrows. But really, what we're here for, the reason why I'm talking, is because of my mom, who just passed away from cancer. Right, so this work that we're doing here is very applicable to real life. My dad kind of talked about all these bullet points, but I talked about the bottom point in red here, where my mom was a very resilient and strong person, and also very dedicated and worked very hard. And one of the lessons that she constantly taught us was that only action counts. So you can say whatever you want, you can claim all these things, but really at the end of the day, only action counts. That was something she sort of drilled into us as like a lesson. And it really got her from, she moved from Korea. My dad didn't say it, but, at the time, Korea is now very popular in K-pop, etc. But Korea was a very poor country post-war. And really, to be an immigrant, I know a lot of people here are immigrants, to claw your way up in America is difficult. And she really did it using this mindset that only action counts. Like, so people like to say they want to do things, and I have this hobby, I want to do it, I want to write a book, and all these things, they don't do it. But take it as yourself, for yourself as a lesson, just in honor of my mom's memory. Only action counts. Something that maybe you have on your list that you never get to, just do it. Okay, that's kind of like what I want to say in honor of her memory. That's what she would have wanted you to do. So I hope that we continue this lecture. Maybe we can have it every year or something like that in honor of her. So just want to say thanks to my my professor at the Hebrew University, Asaf Levy, and to my company Denovai and the venture studio Ion that we're in, and of course to my mom. That's for feeding my son, so her grandson. So thank you, Mom, for everything, and that's it. I know if you have any questions, I know we weren't exactly in the same field, but if you have any questions, I'm happy to answer them or try to. If not, then we, you know,
Yeah, good.
yeah,
Yeah, you using machine learning.
yes,
Machine learning is helping you do your work
yes,
effectively.
yes. So, basically, in the past, the way that, for example, you could design a protein in the past, for example, and the chemical backbone of that protein would be sort of good, sort of kosher. It was okay, but to find the sequence combination that could actually create that, chemical fit was essentially just sort of a question of random searching. Very inefficient, extremely inefficient. Now we have, let's say, a graph neural network that solves that in a second. If you do it 100 times in a second, so time and effort and money is being saved. On a personal level, of course, on the coding side, unbelievable amounts of time are saved using coding tools. Like I know that you should learn how to code actually, but also efficiency wise, it helps a lot. And yeah, everything we do is really tied in with machine learning. There's nothing we don't do isn't in the machine learning. Any data that comes out, we usually are building a model to kind of get extract the essence of it, so we don't have to punch so many numbers. I think everything we do really has to do with AI. Even the physics stuff that we do, we build machine learning models off of it. So I feel like I can't say enough about it, but yeah, pretty much everything. Yeah.
I have a question, Yoshua Berg. You hear me?
Yes.
You first of all, it was a very interesting talk, and you explain very clear, complex things. What I want to ask is about the use of the word hallucinations.
Yes.
We call it when the system is giving you something which is wrong, but here you actually, if I understood right, you just let the computer or algorithm generate things by itself, like
Yes.
go
Yes.
on. And it's not the word hallucination is maybe not a good word for this.
Yes. Yeah.
Because
Yeah.
of the I understand why you use it, but it actually has already a occupied meaning, which has some idea that something is wrong in what you generate. And
Yeah.
here it's not wrong, it's that's wild.
It's true. Yeah, I think you're right. I was confused by that myself when I entered the field. It is confusing. I think perhaps like the landmark paper that started doing this used the term deep neural hallucination or something, and since then we're all using it. So yeah, I agree it's confusing as well. You're right. It's distinct. It's not meant to mean wrong answers, so to speak. But it's meant to be like you the trained model, you put in inputs that don't exist in real life, and yet you get something in the output. So it is distinct, it's confusing, and I really hope that it doesn't cause issues in the future, although, terminology does tend to cause issues. So yeah, it's a good highlight. I forgot to mention something like that.
The second part is very interesting. Already done because I don't see you present the method in detail how you do that.
Yeah. I can't really talk so much details about what we're doing at the company, but in general, I wanted to just highlight some of the big papers and methods in the field for protein engineering. So I would say that those are some of the leading ones. Like these papers are published like now, like they're really new, especially the Minecraft paper was published very, very recently. So they're really up to date kind of like methods. We integrate those kinds of methods and physics-based sort of molecular dynamics methods, which are simulating the protein movement. But yeah, so I just wanted to give a little tour of what type of things that we're using in the field in general. But that's up to date. But it changes so fast. You know, you got to keep an eye on it. Yeah.
Okay, thank you.
Yeah, sure.
Any other questions?
Yeah, one more here.
One more question.
Yeah.
Yeah, like you have the conditions,
Yeah.
right?
Yeah.
The diffusion model, right?
Diffusion, yeah.
But you only consider conditions on one. Or one is on two.
Oh yeah, you can.
I'm saying like, yeah, like not to, and then like the outcome. Basically, you train the model in order to do a lot of things with them, like whether it's to combine them, whether you know what I'm saying, like it's still big knowledge.
Yes. You can condition on multiple targets. You can condition to make it into a certain shape. You can say, I want to condition it to constrain it to diffuse, but within a certain radius, within a certain, you can make it into a, symmetrical ring. You can do a million things. Yeah.
All right.
Yeah.
And if you keep doing that, like in a sequence, maybe you can engineer whatever you like.
Absolutely. There's a beautiful paper that does the diffusion conditioning, and it makes the whole alphabet. They make proteins in the shape of the entire alphabet. Like you can really play with it a lot. Yeah, there's a lot you can do with it. It's amazing. The goal, I think, would be, from a practical point of view, is to condition it to also bind well, also be stable, also be, have favorable characteristics for being in the body, so it doesn't react to the immune system and stuff like that. If all that information could be integrated into one model where you can diffuse the protein and it binds and it doesn't cause an immune problem, and that's like $1 trillion model. That's like
like where you are or where the community is.
Yeah, so I think the whole field is now working on. I think the problem of simply binding, a lot of people have shown it now. So the next move is indeed for immunogenicity, hydrophobicity, all these things that make a drug. You can make a protein binder with these technologies, but that doesn't make a drug. A drug is something that you have to test extensively, that also has to go in a mouse and sometimes also a monkey and then a human and not cause any problems. These proteins do one thing, they're optimized for binding. So that's the gap that the field is trying to close now. So, yeah.
Can you tell us a bit about maybe the model's breakdown?
Yes, of course. Great question. Really good question. So I talk a lot about revolution, right? But of course, there are some gaps, there are some holes. Number one, a lot of proteins actually are sort of floppy by their nature. They don't have a structure per se, at least not alone. So sometimes there's some protein that doesn't quite fold, it's sort of just floating in space there. And maybe when another protein comes or some signal, it'll kind of fold up into something. But on its own, AlphaFold doesn't understand what it looks like in any degree. That's a difficult one. That's like something you have to use more physics models, maybe. There's so that's one thing that really is a big hole, I think, that we really need to deal with. The second thing wasn't a challenge, but now is being solved by multiple groups, is that it's just proteins. We're not only protein, although it's one of the most important things, but also DNA and RNA and small molecules and all kinds of things that are in the body. And so both, there are a lot of groups now that are releasing, let's say, a diffusion model that has all the atoms, not just sort of the protein bits. It has protein and DNA and ligands and all that stuff. And so you can do diffusion of all different kinds of atoms, not just proteins, to get a more realistic picture. It's kind of a gap that's being a little bit filled now, but it's not fully there. But I still think the biggest gap is, the floppy kind of, they call it intrinsically disordered proteins. So proteins that are just sort of floppy by their nature.
Okay, do single nucleotide edits or single amino acid edits?
Yes, that's true. Yeah. Also, so if you want to understand, you take a protein and like we were saying over here, we have like a change in one letter. AlphaFold doesn't understand that change really that much either. So that's also something really important, because people get mutations and God forbid it leads to some disease or whatever. So that AlphaFold doesn't have the resolution also. So that's a good point. Yeah, that's also a really a big gap. But yeah. So either way, it's still pretty cool though. But it's good to highlight. It's important to highlight what it can't do, because there's a gap for everyone here to do something about it, right? So, okay, yes, we finished now. Thank you all for. Thank you very much, everyone. Thank you.